So I understood that some of you are a little bit shocked by molecular biology. So please stop me at any point that you are missing some of the explanations. Wait, we are seeing myself in the, yes. Okay, good. And I'll try also to remind you what is important, because the most important thing is to understand the concepts of how things work. But of course, all the names of proteins and systems is not really relevant. It's just to give you an introduction of what is happening. So today we will talk about the cell cycle and its regulation. And to live and to be and to continue, have continuity, we have to divide and reproduce. So the first thing we have to do is to have a cell cycle that allows cell division. And during the cell cycle, several things have to happen. First of all, we have to copy the genetic material, but also all the cellular components, right? If we have mitochondria, NER, and organelles to make the cell function, they have to be duplicated so they come also to the next cell. We have to grow in size. In most of the cases, we'll see some cases where they don't grow in size to allow the division. And then we have the cells that can be outside of the cell cycle or starting after division to be in G1, preparing for division, where we have cell growth, organelles start duplicating. Then the S phase, that is the synthesis of the DNA, that is what is called S, from synthesis. Then we have the G2, where we check that everything was duplicated, that all the DNA is being synthesized, and that we have the correct signals to continue to mitosis. And then in here, we just start separating everything. So from one single cell, we can have two daughter cells. So for a long time, we have been thinking about this as a symmetric process. But we know now that even in cells that look identical, many cases, the mother cell retains the junk. For example, it was shown also in some bacteria and in yeast that the mother cell will keep all the aggregates and the misfolded proteins and all the things not to pass to the daughter cell. So in this case, the division is not completely symmetrical. We have two exact same cells, but the older one has to keep some of the things to prevent aging of the system as a whole. We have some rejuvenation because the next cell doesn't get all this junk. Also, in our system, we can have cells that are asymmetrically divided. We'll see it also, that can one remain being a stem cell, and the other one can differentiate. And in this case, we can keep the pool of stem cells that will continue to be stem cells and have differentiated cells to generate a tissue. Okay, so we have very basic concepts again. The interphase is when the cell is not dividing and not planning to divide, right? So if a cell is just doing its regular functions, it's an interphase, and this is when we see the chromatin as the spaghetti we talk about in the epigenetic class and genetic class. When the cell wants to get into the cell cycle, it usually has to get the signal from the outside that it's time to divide. And we'll see later on in the class that when we don't get these signals or these signals become unimportant for us, in many cases we get cancer. Why? Because we ignore the environment, if it's appropriate to divide or not appropriate to divide, and we divide anyway. So when we want to start dividing, we start doubling in the size, starting replicating the DNA, the organelles, and then we condense the DNA to allow us packing and cell division. So the first part will be G1. In G1, we allow cell growth, so we accumulate nutrients. We need to know that we have enough energy to go through the cell cycle, because if not, it's not worth starting the process. So we check that we have enough nutrients. We check that the cell grew to the size it needs to grow because before we start synthesis of the DNA. And if we have this signal from the outside that allow us to go on. For example, C. elegans, that this is a small worm. When they don't have enough nutrients, they can become stuck in the dower stage of development. They already have all their cells, but they don't develop fully to be a worm. Why? Because signals are missing from the outside, that they have all the required nutrients to continue with differentiation and with all the development. So it's very important to get the signals, because if not, we waste energy in a cell that will fail and die in the end. And the other thing that is important in G1 is spindle formation, that this will be the ones that help us separate the chromosomes and divide the cell. So this happens in the first stage in G1. Then we have S phase, where we start all the DNA replication, and also the centrosomes that will help us divide the cell. And in here, the most important thing is to prevent DNA damage, because again, we don't want to continue to the next generation, the next cell, with some kind of problem. So the most important checkpoint activation is if we have DNA damage, we don't continue. We stop the cell cycle, we repair, and then we go on. And these are the centrosomes that are at the beginning in one side of the cell, and then they duplicate and move to both sides of the cells. And then it's a mechanical force that will help us separate all the chromosomes and everything in the cell. Then we go to G2. We are supposed to have all of our DNA. Now, we don't know, and it's still like an open question in biology, how we count that we finish synthesizing the DNA. For us, if we look in the microscope, it's very simple, right? We look and count the chromosomes. But how a cell counts? No one knows, right? We replicate the DNA, we expect it finished, but what is the signal that everyone was counted and we can go on? We don't fully understand that either. But we get to G2, and we check that we have all of our chromosomes ready, and the cell is big, there's no DNA damage, and the DNA is fully replicated. Then we can start the chromosome condensation and start opening the nuclear pores. Because if we need to divide, we need to take this and separate the cells, so we don't need a nucleus anymore. And then we go to the Mythotic phase, that this is, I think you have been hearing from high school, and no one really ever remembers what is prophase, metaphase, anaphase, and all the others. But just as a reminder of what is happening is that we have the breakage of the nuclear membrane in G2, and we have the centromeres attached to the kinetochores. And then we start dividing with the prophase, where we align the chromosomes in front of each other. Then the spindles attach in the pro-metaphase, and the chromosomes condense even more, so we can start separating them without causing damage. We have to already start a better alignment, and also we have some coesins. You remember these molecules that hug like a ring the chromosomes? When the DNA is replicating, they hug each other, so things don't get lost, and replication is easier, and we can count. Maybe this is one of the systems of counting better. But at some point, we need to open to allow everything to get separated. So we need also to open the chromosomes at some phase here in metaphase, because in anaphase, we are already starting to pull them apart. And then in the telophase, we start regenerating the membrane, and then the cytoplasm, and then that's it. We finish the cell division. And we have two daughter cells that can be identical or not, depending on the circumstances, depending on the tissue, depending on the organism, depending on many, many things. So how did we study the cell cycle and how we discovered about the cell cycle? And we will start with the very basic methods that were used like 50 years ago that allowed the first discoveries. So many of the things that we know come from yeast, because it's very easy to look at them and see the morphological changes. We see them growing and having a budding yeast and then separating. And also from frog eggs. Frog eggs are so big that a simple microscope can allow us to see the whole egg. So they are very nice to work with and see, and even with very old technology, it was possible to inject things inside of these eggs, not like other cells that are much smaller. And also, in the end, also mammalian cell culture. So what happens at the beginning, we will start with the frogs, the xenopus division, is that we have the fertilization, the eggs go through fertilization, and we have 12 rapid cell cycles that they don't go through the different checkpoints. They go mitosis and synthesis, and in these stages, the cell don't grow in size. We are still using all the information that is coming from the maternal RNA that is around, and all the energy from the egg. Then after this stage, when we want to start also having more polarized differentiation divisions, we start adding checkpoints like G2 to check everything was correctly copied and that we have all the cell growth that will allow us also to start having this asymmetric cell cycle. And some of the cells will go to terminal differentiation, so they don't need to divide anymore. Some will generate larva tissue, and some the nervous system. So what is happening is that at the beginning, the egg in its own maturation. Yeah, sorry. Yeah, okay.
I have a question about the previous slide, because in this cleavage phase, we don't have any G2 phase, so we don't have the DDR thing. We don't have a check of the DNA. So, and these are pluripotent cells, right? Yeah. So how we keep the genome in pluripotent cells? How we see, you know, like which is an error, which is a mutation, which should goes on, and which should not?
So in this stage, we really don't do the checkpoints. But after that, if there is, when things start to slowing down after 13 divisions, and in zebrafish, it's also more or less the same number, then things start collapsing. If there were mutations that are deleterious, or if there are some things that are completely lost, at this stage, we will have like the embryonic loss, but in a very, very early stage, like even not embryo is formed, it's just the ovocyte very, very more or less at these stages, okay, the fertilized egg. But like also in these stages, we are using all the maternal material to check that everything is okay. But this rapid cell division don't have checkpoints. They just continue, and if something collapses, everything will collapse. And it won't continue. So we lose this material so early that we don't have a mechanism yet to understand it, because these are very, very few cells, very, very small, and when something goes wrong, they immediately go under apoptosis, so we don't have enough information to do research on them. But we know that after we start activating the checkpoints, if something was wrong, if there's a mutation, it will be already accumulated. But if it's damaged, it will be sensed, and the cells will, the embryo will collapse.
But what would be the case in iPSCs?
In the what?
Induced pluripotent stem cells.
So the induced pluripotent stem cells, you take a mature, already differentiated cell, then you reverse the epigenetic code to make it more flexible, right? So they now look like stem cells, and then you redifferentiate them. The efficiency of producing stem cells is below the 1%, or now with new methods and some drugs, maybe it reaches 1 to 3%. But it's a very, very inefficient method. And why? Because there are so many barriers preventing a cell to go through dedifferentiation that most of the cells collapse and die. So really reprogramming is so inefficient that it's the outlier. No, it's not the main thing with induced pluripotent stem cells and everything works. No, these are the outliers that for some reason manage to go dedifferentiation. But most of the cells will not go through dedifferentiation and will die because accumulated damage, because they have barriers not allowing to remodel all the epigenetic code, because they will understand that something is wrong and go to the checkpoint activation. So in those cases, the majority will not function. Okay? In here, we really have very little information because of the size limit, but now we have new technologies that maybe will allow us to understand, like the single cell sequencing, also RNA and DNA, to see if there is accumulated damage or not. But before that, we will do the bulk, right? And we will get the statistics of everything. But now, maybe these methods will give us more information regarding these first stages when there is no checkpoint at all. More questions? Yeah. The microphone. Here it is. No.
Okay. Just one question. I didn't get why we don't have checkpoints, because we do have a checkpoint of DNA damage on the S phase, don't we?
In regular cells, yes, we have it. Just during the embryonic development, during the first 13 cycles of development, and this is in this model, but I know it to be true also at least for zebrafish and other amphibians. And we don't know, I think it's also true for mammalian cells. At the beginning, these divisions are very, very fast. We have the mitotic signal, we have fertilization, and we start dividing very fast until we want to start using also, in here you'll see very soon, not the maternal RNA, but the own RNA of the new organism, right? It has a different DNA than the maternal one. So just at this stage, we start using the DNA, our own DNA, to produce mRNA. So at the beginning, we have maturation of the oocyte till it has a very big size, has all the nutrients, and also maternal mRNA, mitochondria, and everything. Then it's fertilized, and we start cell divisions without changing the size. So in here, you see that we are using the same energy that we had. We are using the same cytoplasm. We are not growing. So for example, G1 is not necessary, right? Because in G1, the main thing was to gain nutrients and to gain size and to have all the organelles. So G1, we can skip it. Why do we skip G2? It's not that clear. Because if we have mutations, they will persist. So we start having G2 around the 14th division, but still we are using the energy and the nutrients from the oocyte. Then when it starts already growing and differentiating, we will also add the G1 and the usage of the own mRNA. So for example, when we start the divisions, we are accumulating the maternal RNA, we are using the RNA, and then we have an active degradation of the maternal RNA. It is labeled with some modifications, and we degrade it, also using siRNA, a microRNA, siRNA, that binds to the 3' of the maternal RNA. We degrade it, and we start producing our own zygote RNA. So now we are using our own information to start the development. But at the beginning, the first divisions are just very fast division to have enough cells that after that, they will start having also polarization, and each one will develop into a different thing. But at the beginning, we want to produce cells that will tell them what to do later on. So these are in the first division, and later on, we will start with our own RNA, and also we are starting to close and open some of the regions. So the usage of our own RNA from the zygote is also already dictating what I want to become in the future, which kind of tissue I want to become. So at the beginning, as I said, the experiments were very, very, how do you say, not brutal, but very, like, just let's try to throw everything and see what we get. So they will take like the oocytes of the xenopus, divide them in a centrifuge to get the yolk, cytoplasm, and lipids, and then take the cytoplasmic fraction and put it into the other oocyte. And then they can see that if they put a fertilized cytoplasm, it has the signals to induce starting the divisions of a non-fertilized egg. So there was a signal, a cytoplasmic signal in all these mix of things that was giving the go on. And then they try to find this metaphase promoting factor. There is something that is promoting cell division. And they separated again things and came up that this was a protein. And what they saw is that there is a protein that goes up and down, up and down, up and down, depending the cell cycle, where the cell was in which stage of cell division. And what they saw is that they have two proteins that form this metaphase promoting factor. One that is a small protein that is a kinase that is constant. It is all the time in the cell. And one that they called it cyclin because it goes up and down during the cell cycle. And they form this complex. And now we have a unit that is like the one that knows how to work. It's the kinase. It adds phosphorylation to the substrate. And one that is the regulatory subunit that can give specificity, know whom you should add this phosphate and when and which proteins should be modified. So in this case, we have a complex that can vary during the cell cycle, and you'll see this in a second. So the last experiment confirming how the cell cycle moves forward or if it goes backward came from mammalian cells. We know how to do what is called like hybridoma. We can put two cells together with a method and mix their cytoplasm. So we have two nuclei, and each one came with its own cytoplasm. So for example, if we put together a cell that is in S phase with one that is in G1, we will induce this one, the one in G1 nucleus, to go through synthesis. Why? Because after G1 comes S, and if the cell was prepared enough, or now it has all the material in here that is telling it it can go through synthesis, it will start synthesis. If we mix a cell that was in S2 with G2, the one that was in S won't start G2. Why? Because we have checkpoints. We have to finish synthesizing the DNA. We have to make sure that we have everything ready before we go into G2. So this passage is not that clear. We have to activate certain checkpoints to make sure that things are ready. And if, for example, we put one in G1 and one in G2, again, this doesn't help me at all. I haven't gone through synthesis. I don't have all the genetic material ready. I haven't produced the proteins that are necessary to go through G2. So we will get stuck with nucleus in different stages, even though the cells are mixed and have the same cytoplasm. So some of the information is still retained at the nuclear level, and some of the information is coming from the cytoplasm that can we continue, can we not continue, and it will depend where in the cell cycle we were. Okay? So the cyclins and the CDKs can control the cell cycle in a very nice way. So I won't go through the different names because it's really not important, but what I want you to see is that we have two levels of regulation. One at the mRNA level, we start producing these cyclins at some point in G1. They go up and they then go down. And then we have cyclin E, the green one, that starts at the middle of G1 and decays at the middle of S, and all the others the same, different kind of patterns for when they are necessary. So this gives them specificity when they should be active, but also combinatorial efficiency when they overlap, right? Because this one overlaps with the other ones. So this gives us a mixed specificity for substrates that can be from the red one and the green one, or the red one and the blue one. So we have some variability that is allowed through this expression of these different proteins. And what happened is that they are regulated also at the protein level. So the kinase, the CDK, is not active until it binds the cyclin. When the cyclin is bound, it will give the specificity, the target recognition. Now I have like the one is like the one that can shoot, and the other one is telling who to shoot to add this phosphorylation. So we need both of them to make it active. But also we can fine-tune this through different modifications, such as autophosphorylation, or for example, if we have a cyclin with the green subunit and another one with the blue subunit, they can regulate each other and have a communication between them and how to proceed. And this will depend on the concentration. So we have a balance between these processes, and we can also inhibit their activity if we encounter a problem. For example, we were in the middle of the cell cycle, and we are in S phase, but then we have DNA damage. Then we can inhibit the cyclin activity to prevent continuing with the cell cycle because something is wrong. So we can also regulate them at the protein level to see that everything is going on correctly. So in a schematic way, you can see here the CDK is constant, it's this brown one. And then we start the cell cycle with this red one. It binds, they act together, and then it gets modified, ubiquitinated, and degraded. So now this one is free to bind the green one, and then they act together to promote the S phase. And when they are done with that, this one also gets modified, ubiquitinated, and degraded. So again, the CDK is free to bind the new partner, promote this G2M phase, and get degraded. And then we are recycling the CDK again. And this is how we allow a cyclic, like a clock machinery, to allow us to move through the cell cycle. So it's a molecular machine that really recycles some subunits, and each one influences the other in a way that we can have periodicity. Okay? One of the errors in the cell cycle is one of the main cancer reasons. Yes?
Okay, sorry, I didn't get like how we can have the S and G1 and it becomes S with these mechanisms, because we have the, like, in my mind, if we had the G1, we would have the proteins, the cyclins from the G1, which would be linked to the CDKs, and I didn't get like how it passes to the S.
Okay. So in here we are simplifying the things, but imagine that we are in G1 and we start phosphorylating things that say, okay, let's produce more mitochondria, let's enlarge the membranes, let's do all of these things. So this kinase is activating all of these pathways, but at some point, some of the things that it activated have a feedback on itself. So we have a CDK with, we'll call it cyclin 1, just for simplicity, that activate many things that will allow cell division. So we'll have the nutrients and organelles and many other things. Between these substrates, I will also activate some proteins that give feedback here. For example, phosphorylate this protein. When I accumulate enough of this regulatory protein, we'll call it B, then this one gets sent to degradation. And now, also at the mRNA level, we here also have some transcription factors that will start synthesizing the mRNA for, we'll call it cyclin 2. So now I'm starting to produce this one. I degrade this one. So now CDK is free. So now I move on to the second one. And in this way, we have a regulatory feedback that generates the next one at the mRNA and protein level. I degrade the one that I don't need anymore. I have to kick it out from the complex. And these are depending on various of the different mechanisms that I am activating downstream. So in many cases, it's not a single phosphorylation. For example, I have to have on the cyclin 1 a phosphorylation in 2 places. The first one I get from a process that was activated here, and the second one from a process activated here. Only when these two happen, I get ubiquitination to degrade it. So the signals are coming to allow like a combinatorial way to prevent the cell going through things when it should not go through them, and to get the feedback from various of the mechanisms that have to be fully activated. So when we are done with this one, we degrade it, and the other one is already waiting, because at the mRNA level, we already start producing it. And then we will shut it down with transcription factors that are sitting there, and we move to the next level. So it really works like a clock that needs the other one to be ready for the next movement. More questions here? Okay, so this is one example of a transcription factor. Like I was saying here, we activate some transcription factors that allow us to move to the next period. That is the retinoblastoma. Retinoblastoma is usually is an inherited mutation. Kids with retinoblastoma already have the first hit, one mutation in this protein. That the second one is really easy to get, and this is a transcription factor. So before I move to what retinoblastoma does, I want to remind you of the two-hit hypothesis. That is that to get cancer, we have many, many backup systems preventing the cells to go through the cell cycle. So usually one single mutation won't get us cancer. We need a second hit to move and really impair 2 systems, then the cells can divide without authorization. So in many cases, one of the kids are born with one mutation, for example, in the retinoblastoma gene, and during development of the eye, they get the second mutation, or in retinoblastoma or in another cell cycle regulator, and then they get this type of cancer. So retinoblastoma is a protein that can bind this transcription factor called E2F. E2F activates genes that allow cell growth, and when retinoblastoma is bound to it, it cannot activate these genes. So retinoblastoma gets phosphorylated, for example, by these CDKs, and when it's phosphorylated, it has lower affinity for E2F, so it releases it. And now E2F can allow the program to go through and allow cell growth. If we have mutations or we don't have retinoblastoma, this E2F is constantly active and is promoting cell growth, even though we don't have the regulation of this protein telling it, wait till I get phosphorylated, wait for the signal, and then we have an impairment in cell division, okay? So many of the mutations that we know that are important in cancer, for example, p53 mutation, is one of the sensors and is one of the checkpoint activators that prevent the cell to go through cell cycle. So about 50% of the cancers have mutations in p53. Some of them p53 and other things, but it's the main gene that is mutated in most of the known cancers. Of course, we have for each particular cancer other mutations, but this is one of the main regulators of the cell cycle. And for example, when we have DNA damage, we activate the checkpoint proteins, and they tell p53 that something is wrong and stop the cell cycle. We activate the expression of proteins such as p21, we'll go to p21 in a second, that bind this complex CDK and cyclin and don't allow it to continue working. So if we have damage, we tell the cell, okay, time to activate the checkpoint and call p53. p53 is a master regulator. It starts producing these important genes, and these genes are usually inhibitors of the cyclins. So we produce p21. p21 comes and stocks here in this complex and says, now you are not active. You cannot work until the damage is repaired. Once the damage is repaired, we start degrading p21. In some cases, we have to delete the whole complex and create new proteins to allow us to continue. So it's very important to keep all these signals to allow division only when it's needed, when the cell is ready, when it doesn't have mutations, to prevent all of these diseases that we know are cell cycle diseases. So p21 is one of the most important inhibitors of the cell cycle, and we will see that it's one of the main markers of senescence. If we express too much of these proteins, the cell will get to a state where they don't divide anymore. Even if they get the correct signals to start dividing again, they don't. They just left the cell cycle forever and have had enough of it, and goodbye. Someone else should divide, I am done. So this is one of the main regulators, and it's activated by various pathways, but mainly also DNA damage or some mistakes in the p53 activation, that it could be also hormonal. And it blocks also the retinoblastoma phosphorylation, and it blocks the cyclins, and it blocks in this way proliferation to various interactions with other proteins. Okay, so 1 second. Any question about these proteins? Okay. So again, the names of the proteins are less important. Some of them you will always hear in the literature, like p53 and p21, but the others are less important. So if we have damage, we want to activate the checkpoints and prevent from the cell to entering the cell cycle when it's not ready, when DNA is not complete, when the cell senses that it's not completely, yeah, sure.
Sorry, I was just curious, like, why senescence and not apoptosis? Like, why don't we kill the cell, or why don't we just let it there?
Kill the cell or let it there?
Yes.
So in some cases, if the damage is too much and we don't manage to get rid of the damage, the cell will go or through apoptosis or through senescence. If the cell doesn't understand this, then it will give us hyperplasia. It will divide uncontrollably. But most of the cases, we don't want to waste this cell. We want to let the chance to repair. But if it doesn't manage to repair, then it will have these two options. go through senescence or go to apoptosis. So they will get there, but depending if we pass the threshold of damage, that tells them it's not worth it.
What's the difference between senescence and apoptosis?
Okay, so we will get also to senescence in the last part of the class because it's like a getting out of the cell cycle. But in apoptosis, it's irreversible. The cell dies. It was enough, it's not functional, it collapses on itself, degrades, and it's gone. We don't have the cell anymore. In senescence, we have a cell that is half functional. It's still there, but it's not completely working. It cannot divide anymore. It's also nagging the neighbor cells, telling them, I am sick, take care of me. We'll say it in a minute. And at some point, it also can spread this and damage the whole tissue. So senescence, the cell is still alive, but it's not so functional. Apoptosis, the cell is dead. It's gone. So we don't have the cell anymore. So another thing that I just wanted to point out, because you have been studying more system biology and evolution, and this thing is that we can translate every one of the molecular problems into a system problem, right? We can say that we have input from some signal, some are inhibitory, some are activatory, and then they have their own networks, right? The other proteins that can activate or inhibit or send to degradation and these things. So every problem in molecular biology can also be translated to a problem in system biology, right? Where we have inputs and outputs depending on the network, depending on the different interactions. So don't get scared because you don't know the names of the proteins or all of the processes, but once you learn in which function they are and with whom they interact, this can also give us output of how things work in a more general manner and not one by one. Okay, so in here, for example, if we have DNA damage, it activates this protein, ATM and ATR, two different proteins. And then they can activate different proteins that have different interactors or can activate one to each other. So we can translate this signal into a network that functions together, and depending on the strength of the signal and the different interaction that it's doing, we will have a different outcome. And this outcome can be also tissue specific. For example, in some tissue we don't have, for example, like the example with BRCA1 and BRCA2. Why do we get breast cancer and not skin cancer, right? Specifically in this tissue, BRCA is the main protein helping regulate DNA repair. In other tissue, we have some other proteins that can come and change and do the work of BRCA. But specifically in the breast, the other proteins may be expressed very low. So this system will have variability depending on the tissue, depending on the stage of the cell cycle, depending if it's young or age. But we can translate all of this to signaling a network of the different proteins. Okay? And for example, in here, I just want to point to some very nice experiments that can be done using what we know about biology. For example, this is FACS. You know FACS, sorting of the cells. So what we can do to study individual cells is that we can label them with different colors depending on the DNA content. How much DNA we will have more of certain color, for example, blue. We can label some membrane proteins to see if they are apoptotic, if they are dividing, or if they have activating some proteins. So we label these cells with different colors. So we have a population of cells, and each one can have different signals. For example, some are blue, and some are blue with yellow, and some are only yellow. And then we pass them through a filter and some laser that detects the different colors. So each cell passes through this filter, and a laser reads which colors we have. And then we can have information per cell about different characteristics of the cell. So if we look here at this experiment, we can measure the DNA content. So it's telling me if they are replicating or not, if they have 2N, 2 copies of each chromosome, of if they are in the middle, so they start having more DNA, but they haven't gone through the full 4N. So I have variability there in a population that is going through a cell cycle, and I can measure the DNA content, and I can measure also the incorporation of nucleotides. So how many new bases I was adding. So for example, in here and in here, I have the same amount of DNA, 4N and 4N, but in here I'm still incorporating things, and in here I am done. I don't incorporate new nucleotides. So here I am about to finish synthesizing the DNA, and here I am done. I am not incorporating. So I have in here 2 parameters that I can measure that give me information regarding where in the cell cycle we are. And then, for example, we can label for proteins that are activated or non-activated, for example, cyclin B intensity, one of these cyclins that we were talking about. So in normal condition, I expect it to go up, and then in G2M to have the highest expression, because it's important for G2M transition. But if I have a defect in signaling, I will start seeing cells early on having cyclin B. So this is a very beautiful method that allows us to analyze a population, but not in average what is happening in the population, but where each single cell is in this cell division, or we can measure differentiation of cells, activation of beta cells, T cells, different markers in neurons. So it's a very nice method, and it can give us a lot of information, and it just here was to show us an example. For example, ATR is one of the proteins that signal that there is DNA damage, and when we don't manage to signal that there is DNA damage, we silence this gene, then we start having signals to move to G2M before time, before the DNA is completely synthesized. So this is just more to show you the methods and to show you how, for example, if we manipulate one gene, we can see the outcome, and this helps us understand what is the process, where things went wrong, right? Because we expect maybe to be activated in S phase, but then we see it in the end of G2M, so it's different. Okay, then we'll go through this fast. Let me see how we are with time. Okay, then we have the spindle attachment. We separate the chromosomes, and when things go wrong, we can see these anaphase bridges, meaning that the cell divides, and I still see a little cord of DNA that when the cell pulls strong enough, this will be broken. And then we will have damaged DNA that we cannot repair anymore. We are in the middle of the cell division, and it's very common in the microscope. We can really see them happening. And in the end of the day, we have to exit mitosis and decide that we are done with the cell cycle, and we have our own complexes that are important. And for example, APC is one of these complexes that signals in here that everything is okay. If everything is not okay when we are already in mitosis, the cell will go to apoptosis because it's too late to do something else. And this is one of the most common mutations in colon cancer. This is I think at least 30% of the known mutations in colon cancer will have APC mean mutation. So it's very, very important to control this stage, because if we don't control, we have undivided cells that have already broken DNA, missing pieces of information, and we can get cancer. So to summarize the cell cycle in a normal way, we have G1, where we can check the environment is favorable. Everything, we have nutrients, we have growth, we have all our organelles. Then in the S, we check that all our DNA is synthesized and we don't have damage. Then in G2, we again check, is all the DNA replicated, and is the environment favorable? Can we divide? Do we have enough energy to go through? Because of course, this is also an energy consuming mechanism. And then in N, we have to have all the chromosomes aligned. They have to be separated nicely. We don't have to have bridges. And if we have problems, or someone is detached from the spindle, then we should go to apoptosis. We should not continue, because this is too late right now to fix the things. So again, the most common cell cycle disease, if things go wrong, is cancer. That is, of course, also one of the most common causes of death, not only in humans, but in any animal, not any animal. The naked mole rats and some other species have less cancer, but I brought you some statistics, 1 second here. For example, we usually think that our dogs don't get so much cancer, or our animal mouse, most of them die from some cancer type. So if we look at humans, we have approximately 500 cases of cancer in a 100,000 people population. But in dogs, we have 5,300 dogs for the same size population. So we have developed mechanisms that are better at protecting us from cancer from other species. Also, in our animal models in the lab, many of the mice died from cancer. Okay, so in the end of the day, when we check longevity and we check if we are really improving the lifespan of these animals, we check why they die.
Sorry, I'm confused. It's 5,000 dogs affected by cancer by 100,000
Yeah.
dogs. Okay,
Yeah,
because I was
of
thinking
course-
like there are 5,000 dogs by 100,000 humans.
No,
and,
no.
and
It's-
then I was confused. Okay,
No, no, each one individually. So, but we have our own mechanism. So, just to finalize, so we have, when we get cancer is when we have a cell that does not need a signal to enter the cell cycle. Cancer cells become immortal, and sometimes they reactivate the telomerase. We will see very soon why telomerase is important. And they bypass the checkpoints. For example, they don't have p53, they don't have p21, they don't have retinoblastoma. And they ignore the damage and fail to initiate apoptosis. So they just continue as everything was okay when damage is accumulating in the cells. And another important thing is that the second hit can be epigenetic. For example, we are already born with a mutation in some important gene, but then epigenetically, a tumor suppressor is silenced. It is there, it could be used, but was silenced. And this is one of the things also that we see in aging, that most of the genome gets loose, meaning that we generate mRNA that we should not. We are wasting energy and we have much more noise in our RNA, but specifically tumor suppressors are repressed. Why is that? We are trying to understand that, but a second hit in cancer, it is possible that we don't find the second hit in a genetic screening, but we find that these genes are silenced. So a second hit could also be epigenetic, okay? And again, I just wanted to show you.
I have a question. We always talk about mutations in P53 and the others, and you said something about epigenetic phenomena that can silence genes. Can we also have some kind of mutations in epigenetic level? For example, it was marked by some random causes.
So yeah, when we mean that we have an epigenetic mutation, it's not a mutation really because we didn't change the code of the DNA, but it's a region that usually in these cells should be open. For example, we are supposed to have all the time P53 or SIR6, that is the protein we work with. We see in aging that it gets silenced. So the gene is there, the mRNA should be expressed, but it's not. So something happened at the epigenetic level and not the genetic level that silenced this gene that is important to protect us from disease. So there is no mutation, but there is an epigenetic change that is preventing this tumor suppressor from being expressed. So it can be induced through epigenetics and not through also a mutation, because the difference is that if we have a mutation, let's assume we have P53 gene and we have two copies. One of them came mutated, but we still have the other one. But for some reason, we add methylation in here and we put nucleosomes that are blocking the usage. So suddenly, I didn't have this one. And then epigenetically, I am losing this one. So I am left with no P53 and cells that can divide uncontrollably. So this is what we mean by epigenetic second hit. The sequence is there. And now there are many, many clinical trials with epigenetic drugs. that can allow us to open the chromatin. Now, still, all the epigenetic drugs that we know are non-specific for a certain region, but are specific for certain enzyme. So, for example, if I have some HDAC is histone deacetylating enzyme, and we have many families. They have many, many different targets. They can have targets in the cell cycle, in differentiation, in development, whatever it is. So when I use an inhibitor in here, maybe I am opening the p53 sequence, but maybe I am opening also other regions because they are for this enzyme and not for a sequence. So still, some methods are trying to be developed to specifically open a region. These are more or less used in cancer and more in genetic diseases. For example, one of the things that can be done to open a specific region or to close a specific region is that we have, for example, this region that was silenced. We bring a CRISPR-Cas9. Did you hear about CRISPR, right? We can have a mutated CRISPR that it can bind the DNA, but it doesn't cut it. Because usually CRISPR, what it does, it recognizes a sequence and cuts it, right? But we can have an inactive CRISPR-Cas9 system. We bring the Cas9 inactive in here, and we bring it chimerically engineered with a transcription activator. And now I decide which sequence I want to activate. So I bring through the guide RNA my CRISPR-2, the gene I am interested, for example, p53, and I activate this expression with a Cas inactive enzyme, but that is fused to a transcription activator, allowing the transcription of this gene. So these methods are being used more for genetic diseases where we want to silence, for example, a protein that is dominant negative, that is being expressed, is mutated, and is causing a deleterious effect in the cell. It has an effect that is killing the cell. For example, in Huntington disease, right, we have this polyQ. We have a gene that has many, many repeats that become aggregates. So I can, instead of bringing a transcription activator, I can bring a transcription silencer. And I silence this sequence. So for now, because cancer is so broad and there are so many changes, we are still using like general things that may improve the life. But for genetic disease, we can use these kind of methods. Okay, more questions regarding this? Okay, and again, just to show you that everything can be converted to a system of interactions and nodes of activation. And we will go now to the senescence. That was one of the questions. What is senescence? So at the beginning, high flick. Sorry.
It's I don't know if it is a quick question, but okay. About the role of the MAP kinase in the proliferative thing, because we heard about it like
Of
a lot of times.
kinase?
Yeah, but I don't know where they relate for, like on the CDKs, on the cyclins, on.
It has so many roles, MAP kinase.
Yeah.
Most of the kinases, the problem is that they are very promiscuous. They have roles in so many different pathways that it's very difficult to point where exactly you have it, depending on the cell. But
Yeah, I don't like.
it has many roles in neuron development. It has roles in the cell cycle. It has many roles. Most of the roles for MAP kinase are also depending on nutrition, on signaling from mTOR and all the energetic status of the cell. So part of the control can be also in this sensing the environment is favorable to go to cell cycle.
But then when would they interact with all this machinery that we saw, like in the G1 phase?
In various stages. The thing is that it has many different places where it can interact. And this is the problem many times with trying to do like the interactome of a kinase. They have hundreds or thousands of proteins they can interact and phosphorylate. So for example, for ATM, that is another kinase very common for DNA damage, the interactome was about 3,000 different proteins that can interact in different stages of the cell cycle, repair, or roles simply in chromatin. So MAP kinase is also one of the most studied one, and it's everywhere.
Okay.
Okay, so I don't know.
And another question that I have is that, as far as I know, ever since we have a single-strand DNA in the nucleus, the RPA will be recruited, and then the RPA will bring the ATR. And then how the cell deals with this in the regular replication, you know, because then you need to
During
produce-
regular replication, yeah, the helicases will help, like RPI will protect the DNA, and then we bring together with the replication machinery helicases to remove RPA and to continue. Because of course we don't want to have the single-stranded DNA hanging around, but we need to remove it to copy the sequence. So polymerases always recruit some helicases that can remove also RPA, not only coiling of the DNA, to allow replication to go through. And when it doesn't happen and we have this constant, then we activate ATR. Mainly ATR is activated in the cell cycle, and ATM when there is damage in the cell cycle, and ATM when it's during any other time.
Okay, thank you.
Yes.
The leukemia is also related with mutations in cellular cycles.
I don't know specifically about leukemia, but most of the cancers, the first hit will be or DNA repair enzymes or cell cycle. Why? Because if it's DNA related, it will allow the gain of mutations more easily, and some of them will be in cell cycle proteins or in a cell cycle, then they have uncontrolled. But also in leukemia, there are different types of mutations, and I don't know all of them, but usually as a rule of thumb, that is correct in most of the cases, the main drivers for tumorigenicity are DNA repair and cell cycle regulating enzymes. But specifically, I don't know. Okay, so we'll go now to what happens in the other way, when the cells stop dividing but keep living, as in senescence. So the first experiments were done with cells that were taken from fibroblast tissue, like not an immortalized line, and Hayflick noticed that they have a finite replicative lifespan, meaning that after some replication, they stop dividing. And this was because of the telomeres, right? The telomeres are shortening. Then we said, okay, no more. We have divided enough. Now it's dangerous. We should stop. And of course, we have some exceptions, for example, germline and ES cells or tumor cells that in many cases have an active telomerase. And this gave rise to the Hayflick limit. In many cases, if some cells manage to escape this stage, then they become immortalized. Immortalized and is not the same as completely cancerous, because they have to have a different mutation or additional mutation to become also tumor prone, but they don't die. They can keep dividing forever. So many of the experiments we do in the lab in cell culture are with immortalized cells. Sometimes they are cancer cell lines that were taken from a patient, but in many cases we can also immortalize cells without converting them in cancer cells. Just as an anecdote, I got a book on how do we age and why do we age that was found in the institute, and it was signed by Hayflick himself to the professor that just got to retirement. So I have now my Hayflick book signed by Leonard Hayflick. So okay, what we have is these telomeres start shortening, and then we say it's enough, we cannot continue, let's send the cell to cell cycle arrest. But not just a cell cycle arrest, but a definitive cell cycle arrest. We should not divide anymore because we cannot cope with these very, very short telomeres. So we start sending signals to tell the cells, okay, we are going to like a new differentiated state. It's not the differentiation, but it's a stage where we don't divide anymore. And we start doing some rearrangements also in chromatin. For example, we are less bound to the nuclear lamina, and we form these SAHAV senescence associated foci in the nucleus that are telling us some signaling are changing. Also in chromatin, we are remodeling our chromatin to permanently stop cell division. And as I said before, also we are starting to nag our neighbor cells that we are not happy and we are sick. So normal cells will divide in a logarithmic way. And in here, what we see is the cells that reach senescence, that they cannot divide anymore, and then we stop dividing these cells. So what are the signals that will cause senescence? We have mainly DNA damage, oncogene expression, because one of the senescence main functions is to prevent cancer. So if we notice that something is wrong, the thing that we want to do is prevent the cells from becoming cancerous cells. So if we have DNA damage or telomere erosion or mitochondrial dysfunction that also cause a lot of ROS and DNA damage and epigenetic misregulation, all of this can induce senescence. And then we have the outcome of this, that if there were stem cells, for example, that went through senescence, then our pool of stem cells is gone. We don't have more stem cells to renew and regenerate the tissue. Also, because they are constantly sending signals to the other cells that they are sick, we have chronic inflammation. All the time, we are sending signals that something is wrong, the immune system should be around, and then we have chronic inflammation in tissue that start accumulating these senescent cells. And also, we have some molecular mechanisms that are changed, such as proteostatic dysfunction. We have loss of proteostasis, and we have imbalance in nutrient sensing, and we start having system problems. So usually what happens is that we have some upstream signaling and downstream signaling to allow cell division. So first, if we have a mitogen signal that tells cell go and divide, and a downstream signal that says everything is okay, we go to proliferation and the cells divide normally. If we don't have these signals, then we go to GO arrest. Cell cycle arrest, they simply don't divide, but they are not senescent. If we get just these downstream signals, we immediately read that something is wrong and we go to apoptosis. No, why I am getting signals to divide when nothing is ready to divide? And in the other case, if we get the first signals to start the cell division, but then we see that everything is wrong, then we go to senescence. I should not divide because some of these signals are missing. And again, this is some of the pathways that we know are activating in development to induce senescence. For example, in the placenta, we don't need it anymore. We want to get rid of it. It induces senescence, and then it is degraded after birth. In adulthood, we have more acute form of senescence that becomes chronic, and then it starts damaging the tissue. So senescence could be good in development for some reasons, but then if we pass this threshold, then it becomes very bad for us. So this is one of the examples I was telling you yesterday that are the antagonistic pleiotropic kind of mechanisms that were evolved in our organisms, but in many animals. Because we try to prevent oncogenic events, we don't allow the cells to divide. So when we are young, we are preventing these cells from accumulating and giving us cancer. So for example, if we prevent in a mouse senescence, it will die from cancer very, very young. But if we prevent senescence later on, they will live longer. And we'll see these experiments in a minute. So senescence is controlled by the most important tumor suppressors, p53 and retinoblastoma. And as I said, mice that do not have senescence die very young because this is the main function. So again, senescence will help us when we are young, preventing cancer. But when we accumulate too much, it will kill us faster. And this is one of the examples of the antagonistic pleiotropic mechanism. So we think that aging is also the consequence of this decline in the selection of the cells at the cellular level. So when we have too many of these cells, then we have too much damage that cannot be get rid of. And then we will have also senescence, and these senescent cells will start accumulating also because the decline of the immune system. because also the immune system have senescent cells and cells that went apoptosis, and then they cannot get rid of these senescent cells. We don't want them too much in the tissue. So in our lifespan, many things have changed because we were living until, I don't know, 30 years of age, 40. Some people may reach, very rare cases, 65. And we didn't see all the age-related disease. There is no evolutionary advantage to prevent aging related diseases because we already have passed our genes on, and whatever happens to us later on in life, it's not important, right? So all the age-related diseases that came with the modern medicine and with this increased lifespan cannot be prevented. So all these senescent cells and their accumulation is new to us in evolutionary terms because people didn't live that long. And the organism, after we have already passed our genes on, couldn't care less if we get age-related diseases. So this is part of what we see, this reduction of fitness with senescent cells. So it was selected for tumor suppressor, but after a long time, we got the effects of having too many of these cells because they start sending signals to the tissue, starting inflammation. And just let me go to some of the nicer examples. So in here, for example, if we have one senescent cell in the tissue, it may not cause a lot of damage, but it starts nagging the neighbors with the senescent secreted proteins, that they can start converting cells that are a little bit far away or neighboring cells into senescent cells. They are less functional. And then we have too many of them that we are starting to damage the tissue and the tissue architecture, and then we are calling for fibrosis, we are calling for the inflammation, and we are causing more damage. So usually the immune system should come and get rid of these senescent cells. So again, when we are young, we have less senescent cells because we haven't accumulated too much damage, and we have a good immune system that can get rid of most of them. As we get older, we accumulate more damage, and then we have more of these senescent cells impairing all the neighbors, and we have less immune cells getting rid of them. And just as an example, we can also have senescent-like cells in cells that don't divide anymore. For example, neurons and heart tissue, muscle, are cells that don't divide anymore. So of course, they are already not going through the cell cycle, but they can go to senescence-like process by nagging neighboring cells, sending these senescence-associated proteins, secreted proteins outside and disturbing the environment. So for example, this is again a paper using senescence and the immune system in a system way, try to understand the fitness between the immune system and the amount of senescent cells. So if we take how many new senescent cells we have and how much are removed by the immune system when we are young or when we get older, then we can get the parameters of how many senescent cells accumulate in the tissue. And what they did is they prepared this model, and then they validated this in a mouse. They could really see the accumulation of the senescent cells according to this mathematical model, that these are things that maybe talk to you a little bit more. Another important thing that we see with these senescent cells is that if we get rid of them in old age, mice can be rejuvenated. These are the same age, and in this one, they got rid of the senescent cells. And this is a very cool system, and I'll show you what is the molecular biology behind it. So what they did is they expressed this protein, I'll talk about it in a second, with the promoter of a senescent induced protein. P16 is very similar to P21. So it gets expressed when we have a signal to send the cell to senescence. And they add this fragment of a protein that is called FKBP. FKBP can dimerize when we add this drug, the AP, we'll call it in here. Then they add caspase-8. Caspase-8 is one of the proteins that send cells to apoptosis, but to be active, it needs also to dimerize. So one caspase being expressed by itself, it doesn't do much. And they add IRIS GFP. IRIS is internal ribosome entry site. Do you know what it is? No. In a second, we'll go through. And GFP, just to see that it's being expressed. So just a brief note in here of what an IRIS is. most of our RNA have a cap to allow translation, right? So the ribosome recognizes the cap and starts translation. When we have, for example, a viral infection, the virus, many times they don't come with a cap, or we start degrading the cap of the virus. So what happened during evolution is that they develop a mRNA that folds itself, and the ribosome can recognize it and translate the viral proteins, bypassing our attempt to get rid of the virus and removing all the cap-dependent translation. Also during evolution, with this co-evolving, many apoptotic proteins and things that need to be expressed under stress conditions have an iris element. So this is cap independent. Why did I add an iris here? Because we want to have a single mRNA being expressed. In here, I express the GFP, just telling me that the system is working and my protein is being expressed. And in here, I have my other system. So this is just to help me know that when I have senescence, I'll have GFP. So now we have a system that when I add the drug, it activates caspase-8 and sends the cells to apoptosis. Any question now? Okay. So what they did is to measure with and without the AP in the transgenic mice that have this system, and they can see that these mice with the AP and the system, they can live much longer than these mice. They also chose a model that have premature aging, so the experiment is not as long as 2-3 years, but they have some premature aging, you can see here, and they are living longer when we manage to remove the senescent cells. So when a cell is starting to become senescent, it starts expressing P21, so it's also start expressing this cassette with the caspase-8. We give the drug, it dimerizes, and it kills the senescent cells. And just by removing the senescent cells in old age, the treatment starts around 12 months, then we can have a mice that look much younger than the one that accumulated senescent cells. So through various parameters, so here they started with the clearance at 12 months and start measuring some of the parameters at 18 months. But in most of the parameters, these mice live much longer and have a better cognitive performance and less inflammation, and overall were very, very healthy compared to the ones that could not remove senescent cells. So this is really telling us that if we remove senescence from the beginning, mice will die from cancer very, very young. But if we remove them later on, when they accumulated already, then we have longevity and less disease. So, the important thing is that, of course, we don't have a cassette with these things inside of us, but there were some senolytic drugs that were discovered that have a very similar effect that can affect specifically and kill senescent cells. So there are several experiments going on with senolytic drugs that when they give them to mice without this genetic enhancement or genetic improvement, they will go through senescence, and then these mice will live longer. Yeah.
Okay. In these experiments, because this is like saying before and after, these rat look like the other?
So these ones are, no, they don't look like the other because in this one, they use an accelerated model of aging. So these age a little bit faster, not in 4 weeks, but they age by 6-7 months. So they really, really look very old by the time of the experiment, 18 months. Because mine should be able to live 24 to 36 months, depending on the animal facility. No, it's also important.
But if you like apply those drugs to the mice that is already old, it's not going back.
It prevents, sometimes in some parameters, it does have rejuvenation, not a complete, like now it's a 3-month-old mice, but it can have some rejuvenating effect, and it decreases also the aging. Because also what happens in aging is that it's not a linear process, right? We can see also in humans, right? When you reach the age of 70, then from 60 to 70, you might be very healthy. From 70 to 80, you might be okay. From 80 to 85, the acceleration of the aging process is much faster, so it's not linear. So again, if you prevent in some of these points the aging, some of them will continue with a normal aging, but it's more fast, than the ones that are preventing this, and then they look much younger. So the difference here is also the animal model. In here, the mice were not accelerated aging. They have a different model. And again, with senolytic drugs, we think they target the cell cycle. We don't completely understand them. People that work with senolytic drugs, they will tell you that there are no side effects, no nothing, but of course we don't know yet because we don't understand the mechanism of action, right? To really understand it, we need to know all the substrates and all the enzymes that could be affected by the senolytic drugs. But at least in animal models, they have a very promising effect with various different senolytic drugs by preventing disease and allowing the mice to live longer. How exactly they work? Not fully understood, and also it's very complicated because every chemical that we add, it can be metabolized to so many other things that it's really, really complicated to try. We don't know how aspirin works, right? We still don't know, and it has been used for, I don't know, 70 years because it can be metabolized to so many other things. So in drug discovery, this is one of the barriers. Of course, if it works, we keep using it, especially if we have something that is very bad. But for example, in clinical trials, it's not very easy, but it's easier to get a drug approved for cancer, especially cancer that is very, very fast, for example, glioblastoma, because the patient have no choice. In 3 months, they may die. But just to have a drug approved because you want to live longer, it might not get a fast approval, right? It needs much more research to be available. So the last thing that I wanted to talk about is cell cycle reentry. Yes, sure.
They evaluate the effect in the next generation after inducing this treatment with this drug?
Usually in these experiments, they start the treatment at 12 months, and these mice are not using breeding because usually the breeders are, you keep a separate cage with your mice that you breed and the ones that you do experiments on them because you don't want to have the influence of pregnancies, hormones, and other things. No, things are already very complicated. So I don't think no one has, they have measured fertility, and fertility is improved, but no one continues to the next generation to see the effect because it's a different experiment. Usually you separate these experiments unless you're studying fertility. And fertility was improved in some of the cases, but the progeny and how it was affected was not studied. Okay, more questions on this? So this is the last part of this class, and it's shorter because we know much less about it. And this cell cycle reentry in cells that should not be dividing. For example, in neurons. Our neurons are terminally differentiated. They don't divide anymore, right? But in some cases, we see an attempt to re-enter the cell cycle, but these attempts don't go through. They don't divide. But in many cases, they get to synthesize new DNA and in a way of try to deal with stress. So in many cases, we see that cells, neurons that are going under stress, DNA damage, oxidative stress, some drugs, they will start synthesizing DNA, and then we generate aneuploidy or hyperploidy in neurons. We can see now with sequencing that many of our neurons, for at least I think 3 to 5% in some experiments from years ago, have not a correct number of chromosomes in them. So have sometimes some extra DNA, some extra chromosome. This can also happen in cell division, but this could also happen because we start the synthesis and then we don't go on. If they try to go on, they will go through apoptosis. what we think is happening is that these cells are trying to repair the DNA. Because the cells are not cycling anymore, we have completely lost the capacity to do homologous recombination because we need the other strand of the DNA, so we don't even express these enzymes anymore. So one of the things that we think may be happening in this cell cycle reentry is an attempt to have more choices for repair. So for example, it also depends in which stage the cell tries to get into the cell cycle again. If these are immature neurons, they are very sensitive and they will immediately die. But if we have mature neurons that are already differentiated, have all the connections, and losing them, it's more difficult for the cell because we are losing also all the connections that I already made. So they are more tolerant to this re-entry of the cell cycle. They will say, okay, maybe we can continue with a little bit more of DNA and live for many, many years, while younger neurons that are still migrating, they will not continue and will die very fast. And for example, if we do an experiment in neurons and measure how many of them go, this is the DNA levels, so 200 is 2 copies, and as we move towards the 400 or more, is more DNA that is being generated. So if we add, for example, a topocyte that is some compound that induces DNA damage, it can also be used to see the DNA in the gel when you run it in the lab because it intercalates the DNA, and this is what generates damage. We can see that the neurons start producing more DNA, and we have more cells with more chromosomes, right? And some other drugs can also induce, for example, amyloid beta is not a drug, it's one of the proteins that it's generated in familial Alzheimer's disease. They can also increase from the control the amount of cells going through cell cycle reentry. So this is not completely considered a disease. It's something that we see. But for example, in Alzheimer's brains, we see 10% cells with aneuploidy, while in normal brains, we will see 3%. So this is something that is happening in more when we have some kind of stress or some kind of disease, this cell cycle reentry. And just to summarize all the things that we saw today, the cell cycle is super important also to generate new organisms and to generate all the tissue that we have and to prevent disease. So the regulation has to be very, very tight. When regulation is not tight, then we can have the different disease of the cell cycle. When things go wrong, we can have senescence, cancer, or post-mitotic cells re-entering the cell cycle. And all of this will have an effect on the health of our cell and the organism. And this is just like now that we have methods, we can map, for example, the different amount of mutations or hyperploidy or other things that can allow us to understand what has happened in the history of each organism, right? What is happening and where it is going with and without the DNA damage signaling. And that's it, I think. So any more questions?
I have a question about the senescence, and because one of the graphics you show showing like the progression of how long does it take for a cell line to get to senescence, and I think it was
at the
about
beginning in the
yes, it was more to the beginning.
It's about 50 passages in here.
Okay.
The first one.
That one.
In here, no.
No.
One here.
And how because if you have like a limited amount of divisions you can do before the cell stops dividing.
Yeah.
How can we continue to live like for 50, 60 years and dividing the cells? Do we have like a lot of because the exceptions are very are not so many. So
Yeah.
early embryonic
Let me see if I
embryonic
have it
cells and
here.
germline.
Yeah. So this is for cells that we have in tissue culture, but each cell has a different lifespan. Let me see if I have the slide in here because it's a very interesting. So there are cells that divide really fast. For example, in our colon, in our skin, these cells divide really, really fast. What we have in this tissue many times is a pool of stem cells. So again, some of the cells that don't go through senescence are stem cells. So for example, if we draw how the colon cells look like, we have villi, right? They have cells. And in here we have different cells.
Okay.
And they mature as they go in here. And in here, we have a pool of stem cells. So they regenerate themselves, and then they produce asymmetric divisions in these cells that will be differentiated. So in here, we have a very important pool of stem cells producing these new cells. Okay. In many tissue that has very fast divisions, we will have a pool of stem cells that when it gets exhausted, then also we lose the capacity to regenerate the tissue, for example, also in muscle or other tissues. Some cells then don't divide at all, like in the brain. Right? So this doesn't mean anything for them regarding how many divisions we have, but they can get damaged in any case. So one of the, not experiments, but one of the brilliant ideas that someone came to see the life of the cells is that there were nuclear tests in Las Vegas during some period of time. And this changed the amount of radioactive material in all the people that were born or alive during that time. And then they could measure the half-life, right, of the decay of the radioactivity. And in this way, they could map from people that were born at that time how many cells divisions they went and interchange of the cell life. So some tissue that regenerate very fast have a lot of stem cells, and some of them have telomerase, right, that allow still some cell division, but mainly stem cells. It was also, there were some attempts when the telomerase was discovered, it's like, oh, we found the fountain of youth. And of course, when you overexpress telomerase in the tissue, you get cancer. This is what you get, right? Because we have other limits. This is not the only thing. But mainly the tissues will regenerate depending on asymmetric cell divisions with a pool of stem cells. Other tissues that they don't divide so much, they have also a much smaller pool of stem cells. For example, in the brain, we have neural precursors in the hippocampus, but they produce very few cells that they cannot regenerate the whole tissue. We can generate some new neurons now and then, but not a lot. In other tissue, we have a very good pool of stem cells, but usually this is the main solution and not a slower cell division. Okay, more questions? No?
Can you recap the causal reasoning behind senescent cells in young animal models being important to avoid cancer? Because I got a little confused.
So why they are important as we are young and
when they are young, yes.
Okay. So what we have to imagine is the amount of damage versus how bad it will be for us. So for example, if we have cells that are dividing when we are young, and some of them become cancerous, then this will kill us very fast. But then if the checkpoints are activated and we, instead of having a cancerous cells, we send it to senescence, then we don't get cancer. So statistically, we have some of these cells, like I am inventing random numbers, but every year we will have, I don't know, 10 of these cells or 100 of these cells. So as we start living longer, some of them will get killed by the immune system and some will remain there. So let's say we should have 50. But then the next year I have another 100. And then maybe 50 get killed or only 70 because the immune system is starting also to decay, and this way. So when we are young, we have few senescent cells that are preventing me from dying from cancer. And because they are few, they're nagging to all the other tissue. Like if you see this slide, this one. Let me see if you, okay. Okay, so senescent cells are constantly sending signal to the neighbors, I am not okay. And then they, depending on the length of this nagging to the neighboring cells and how much signals they get, they will be converted like zombies to be a senescent cell. So if we have few cells and we got rid of half of them, then this nagging maybe is generating a few cells, but maybe the immune system still can cope with them. But by not having the senescent cells, for sure, I will get cancer at some point because I'm not getting rid of these cells that should activate the senescence pathway when something is wrong. Because I have two options, activate apoptosis, go through senescence, or becoming cancerous. So when we are young and have few cells, we can cope, we don't have too much nagging, we don't convert zombie cells around us, we are okay. As time goes by and we continue, then we have, I don't know, now 1,000 senescent cells, but now they also have the effect of affecting the neighbor cells. They become senescent because of intrinsic damage, but now they also are affecting the neighbor cells, and the immune system is weakened and cannot get rid of them. So now the damage that the senescent cells are doing is more than the benefit that they were doing by preventing cancer, because they are inducing tissue atrophy. So if we prevent senescence in this stage when we can still cope with the few senescent cells, then for sure we'll get cancer. In here, if we kill them, we still have some chances of getting cancer. It's not that the cancer option disappeared, but they were doing so much damage by themselves, by converting other cells, by damaging the tissue, by doing pro-inflammation all the time, that they by themselves become more dangerous than the chances of getting cancer by itself. So the tables are turned in this case.
I understand the second phase, when the abundance of senescent cells and the weakness of the immune system makes the accounting go towards death. But in the first case, what you have is that the cells, simply by turning some of the neighboring cells senescent, it turns cells which would be cancerous senescent, and they are.
No. What will happen is that they by themselves become senescent because of a reason, right? Why
Yes.
do they become senescent? Because I have damage.
Yes,
And I have damage, and my options are I can die, and then I get tissue degeneration, I am losing tissue. I can stay in the tissue and do half my job because senescent cells still have some function. They are part-time jobs, and like someone in retirement that still comes to half job and complains a lot. So they are, but they became senescent for a reason. They were in the verge of becoming cancerous cells or go through apoptosis, and then they decide, like, okay, I can become a cancer cell, I can go to apoptosis, or I can be a senescent cell. If we remove the capacity for them to become senescent cells, we increase the chances for them to decide to go to this pathway, to become a cancerous cell, because we are blocking the, let's say, P21 or P53. So
Okay, so it's not the presence of senescent cells in young models.
Exactly.
The presence of because as now you explain it, it seems to me that the presence of senescent cells is more like an evidence of absence of cancer, because
at the end of the day, just as a marker, also senescent cells can induce cancer in neighboring cells. They can activate oncogenic pathways, so they can do that. But forget about that for now. They by themselves could have gone through different pathways. Yes. And they go through senescence, preventing them, most of them, from becoming cancerous cells, from dividing without control. So in the young, when we remove the capacity to do senescence, we are allowing them to choose the oncogenic pathway.
Okay.
But after a while, we accumulate too many of these cells, and then they by themselves are becoming a burden to the tissue, and by themselves they also can induce cancer, because they can send oncogenic signals. So the mechanism is different. One is we are preventing the cells from choosing cancer, and second, we accumulate too many of them that they are, like if everybody was retired, only 10% of the population was working, so nothing functions anymore.
Okay, I think I understood, because I was going away in thinking that there being cells in the tissue which are in the senescent state increases the health of the tissue in general by preventing somehow the appearance of cancerous cells and the development of tumors.
Yeah.
But it's not that way.
It's not
So if instead of preventing that pathway the cell can go through of becoming senescence, you had some mystical mechanism of always removing every senescent cell that appeared, you wouldn't increase the rate of cancer. Because
exactly,
okay.
but this of course again is impossible because as also in cancer, these cells express proteins that are intrinsic, right? The main problem with cancer, now I remember when I was studying biology, we were told, okay, we will solve the AIDS problem and viral problems much faster than we will solve cancer problem. Because when you bring an exogenous protein, then you can recognize it. But cancer cells express our own self-proteins. So just to find them and to target them specifically without targeting all the other tissues is more complicated. So again, we try to get rid of the senescent cells with some efficiency, like in this model. It's a very nice paper by Yuri alone. So we can get rid of some of them. I think I have it. I was looking for this for. No, okay. But we have a trade-off all the time. And in this trade-off, at the beginning, we want to not get cancer. But in the end of the day, we get this deleterious effect. And this is the most simple example of antagonistic pleiotropic effect in aging. We have other examples, the things that give us a fitness. For example, if we are very good runners and we have a very good metabolism that spends energy, it can get us to get more food and be very and reproduce more. But in old age, our body is maybe used to too much energy that I cannot acquire anymore. So in old age, this thing that gave me fitness now is affecting my fitness. So this is the antagonistic pleiotropy, and senescence is one of the examples that something that was good for me, but then it becomes bad for me. Okay? Good? So, coffee, cake, everything there. I don't understand this one. Yes.
I was wondering about cell culture. Some lineages that are immortalized and are used for. Do you want to